Hello and good evening. My name's Tom Stoneham and I'm Professor of Philosophy at the University of York. And it's my delight here today to be introducing an online event in York's Festival of Ideas. Today's event is in a different format from how the Festival of Ideas has happened before, but we continue to aim to enhance York's reputation as a city of ideas and innovation through offering the highest caliber public events. The 2020 Festival of Ideas has over 40 online events like this, offering an inspiring programme for all ages. Thank you for joining us today, and we hope you enjoy the adventure we're about to take you on. Today's speaker is Dr. Phil Ball. Phil is a freelance science writer. Previously to that, he worked for 20 years at the science journal Nature. First as an editor for physical sciences, which covered everything from biochemistry to quantum physics and materials science, and then as consultant editor. He himself is a chemist by training, having a BA in chemistry from the University of Oxford and a PhD in physics from the University of Bristol. And he is a fellow of the Royal Society of Chemistry. He's the author of many popular books on science, and today's presentation will be about his most recent book, How to Grow a Human. This book is an investigation of cells, cell growth, and how those relate to life more generally, and also to our nature as human beings. It's a fascinating book, a book which has many aspects, a lot of science in it, but also interesting reflections on how we think about science and how we understand science, and how science, what we say about science, relates to the stories that we tell about ourselves to understand ourselves and our place in nature. It's a book of popular science, but it's also a book of philosophy. So it's my pleasure today to introduce Phil, who is going to give a presentation, and then at the end, he and I will have a brief discussion. So over to Phil. Thank you very much.
Thank you very much, Tom. And welcome everybody to this online experience and this very ad hoc experience. You may hear some slight sounds of an online trumpet lesson going on in the background below me, but that'll add to the experience, I'm sure. Now, I want to begin straight away by sharing my presentation. So let's get this up and running. There we go. So this is the book that Tom has just talked about, and I want to begin here. These are neurons. If you've never seen neurons before, I hope you'll agree with me that they're actually rather beautiful. So these are the nerve cells in our brains. But these aren't any old neurons. These happen to be my neurons. And you may wonder, how can that possibly be? How can this bit of my brain be on a microscope slide if I'm still here talking to you? Well, the answer is that although these neurons are just like those that are found in my brain, they weren't actually taken from my brain. They were grown, and they were grown from a little piece of my arm, from my shoulder, in fact. And that happened about, I guess, 2 or 3 years ago now. And it was the start of my journey to understand one of the most extraordinary discoveries in biological science that's happened during my lifetime, and one that might, among other things, ultimately end up extending that lifetime, or at least, if not of mine, then perhaps of some of you who are younger than me. Because in a nutshell, we've discovered that our cells, our flesh, our tissues, are much more versatile than we had imagined. And it's probably not too much to say that we've discovered that every cell in your body, every piece of you, could potentially be transformed into any other piece of you. And to take that notion to its ultimate conclusion, it seems at least possible that any fragment of your flesh probably holds within it the potential to become another entire organism, another person. Now, to seriously embrace that idea, I believe, is to recognize some real challenges to our sense of self and identity, to reconsider what it means to be human, or in the words of the American cultural theorist Susan Merrill Squire, to replot the human. And I'm going to talk to you about what that replotting might look like and some of the places that it might take us. And some of those places are exciting and inspiring. Some could be challenging and disturbing, socially, ethically, and even philosophically. Let's start here with the cell. So this is what all our tissues look like. We learn at school that we're built up from cells. That cells are our basic building blocks. But cells aren't just passive bricks. They're living entities in their own right. After all, each one of us began from just a single one of these, a single cell, a fertilized egg called a zygote. And here it is. And we're told that this cell replicated to make 2, and then 4, and then 8, and then before long we have a whole little ball of cells, an embryo, that became us. But right away, our cellular nature is more ambiguous than we might have thought, because it's not simply the case that this embryo just goes on getting bigger and bigger until it starts looking like a sort of rough version of a human body. What most of these cells that you see in this image will become is not us at all, but the housing for us. This is how we begin after several more of those steps. This is what we've become. You see this circular entity, which is a whole conglomerate of cells. What you're seeing here is called a blastocyst. So it's an embryo about 5 or 6 days after fertilization. And you see that it's now got some structure to it. It's not just a blob of cells, but this is what might be the surprising part, that what will become us is just this kind of mass, this little blob of cells on the clinging to the wall of this structure. All the rest of it is going to become the housing, the life support system for that little blob of cells. So the cells in this inner mass can develop into all the tissue types in the body, into blood and bone and muscle and skin and kidneys and nerves and so on. And these are called embryonic stem cells. And they're said to be pluripotent, which literally means to have many powers. They have the power to become any tissue. If you fixated on any one of these cells and watched it divide and divide, then you would see that the progeny of it gradually become a specific tissue type, neurons, say, or skin cells. And that genealogy of a cell is called the cell lineage. And the process through which generations of cells become specialized for particular jobs is called differentiation. So these pluripotent embryonic stem cells differentiate to become the specific tissues of the body. And this happens by degrees. So after the embryo has implanted in the uterus, this clump of cells will, first of all, develop a kind of fold or groove in a process that's called gastrulation. And that groove is called the primitive streak, and it marks the location of what will develop into the spinal column, the central symmetry axis of the body. And this happens around 14 days after conception. And this formation of the primitive streak is therefore seen as actually a rather arbitrary, but nevertheless a developmentally significant marker of personhood, because after it has developed, an embryo can no longer split into twins. It's going to become a single person. And so, for better or worse, and essentially for pragmatic reasons, which I'll return to, the formation of this primitive streak defines the rule that guides all embryo research in this country, in the UK, and also in many other countries, that it's forbidden to experiment in a laboratory with embryos after for any longer than 14 days. So this is the basic limitation on all embryo research at the moment. Now, it's this layer of embryonic stem cells that is going to that is kind of separated from the inner mass. This is what's going to become the fetus, and that happens by degrees. So first of all, these cells differentiate into 3 different types, called the ectoderm, literally the outer skin, the mesoderm or the middle skin, and the endoderm or the inner skin. And these turn this inner cell mass into a sort of layered sandwich of cell types, which is the very earliest intimation of a body plan. So the ectoderm cells are the ones that will develop into skin cells and nerve cells. The mesoderm becomes the internal organs and tissues like the heart and muscles and blood. The endoderm becomes the gut and the lungs and the liver. So eventually these things progress further and further, and these tissue structures start to fold and become eventually, by degrees, something that looks a little more like a developing embryo attached via the umbilical cord within an amniotic sac. And that's the kind of stage of development that we're more familiar with. Now, here's the curious thing, though, because every time a cell divides, its entire genome, all the genes that it contains, are copied so that the daughter cells have them too. So what distinguishes a skin cell, say, from a neuron isn't what genes it contains. The distinction is in what genes it uses, which genes are active. And by active, I mean that the genes are being turned into proteins that they encode, and it's these proteins that actually orchestrate the biochemistry of the cell. So a gene is active if the cell's molecular machinery is transcribing its information into, first of all, into RNA molecules, which are then in turn used to make the respective protein. So that's what I mean by gene activity. And so cell differentiation is a progressive shift in which different parts of the genome become active or full silent. So some genes become switched off or on as that process develops. A pancreas, say, needs to activate its insulin producing genes. And the question of how cells acquire their particular fates, which lineage they become part of, is therefore a matter of how these genes are switched on and off. And this process is said to be epigenetic, which simply means that it involves some level of control over and above the genome itself. How it happens is a very complicated process that scientists are still working out. And here's an illustration of some of the things that are involved. For example, we know that there are enzymes that attach little chemical tags to some genes, little molecular groups that mark them so that they are going to be ignored or silenced, so they become inactive. Another kind of epigenetic program is related to how this long stretch of DNA, the genome, gets folded up and packaged inside cells. So some parts of it might be bundled away beyond the reach of the enzymes that actually transcribe it into proteins. And so those genes too will fall silent. The key point here is that, first of all, a genome regulates itself through this complex dialogue, this network of interaction between the genes that it contains. And secondly, and perhaps this is the most important thing, this dialogue is only played out as an embryo develops and as cells talk to one another. Any given cell in an embryo only knows what fate it should acquire because of the biochemical signals that it receives from cells around it, which, crudely speaking, tell it which part of the body it should become. And I can't stress this enough. The point is that the development of the embryo into an organism, into a person, isn't just a straightforward readout of a plan encoded in DNA. It depends crucially on this dialogue between cells. So if that goes wrong, then development can go wrong, even if there's nothing amiss in the genes themselves. Of course, if there is something amiss in the genes themselves, then that too, if there's a mutation, for example, that stops a gene functioning properly, then that too can disrupt the signaling between cells and disrupt the developmental process at some stage. As scientists began to understand about cell differentiation in the mid-20th century, the developmental biologist Conrad Hal Waddington thought up a powerful way to think about it. He spoke about an epigenetic landscape. And the idea here was that early on in the process, a cell starts off in the uplands, so it's like this little ball up at the top, and then it follows a path down through the valley, and it could follow several paths. And each time it comes to a sort of branching point, it has to take a choice which route to follow. And each time it does that, the cell becomes a little bit more specialized and loses some of that pluripotency that it had to begin with. And it starts to become, to specialize into a certain tissue type. And the thinking was that this is a one-way process. So once a cell lineage has taken a particular choice at a branching point, that's it. It can't go back uphill and change direction. And some researchers actually wondered whether cells might lose genes as they differentiate, but it was later thought that it's more likely to be just a disabling of the genes, but a permanent disabling of them. And that was the view that prevailed right up until about 12, 13 years ago. In 2007, a Japanese biologist named Shinya Yamanaka at Kyoto University reported that he could convert differentiated human cells that have acquired a particular fate directly back into a stem cell-like state from where they could grow into entirely different types of tissue to acquire entirely different fates. And it turned out to be surprisingly easy once you knew how. What Yamanaka and his colleagues did was to use viruses to inject genes into the mature cells that have developed into a particular tissue type. And they injected the genes that are highly active in the embryonic state, in embryonic stem cells. And they found that simply doing that, simply getting those genes kind of working again, if you like, by adding more of them, that was enough to turn the cell back into a pluripotent state. And so these cells became known as induced pluripotent stem cells. And Yamanaka was awarded the Nobel Prize in 2012 for his work. This was just 5 or 6 years after this discovery, which shows really what an extraordinarily important discovery it was, and how quickly that was realized. And there are medical implications about the discovery, and I'm going to come to those shortly. But I think the real breakthrough here was conceptual, because the work showed that our cells and our tissues are actually more plastic and more versatile than we imagined, and that Waddington's landscape isn't necessarily a one-way process. We can go back. So these induced pluripotent stem cells can be grown into any tissue type, perhaps even into entire organs like a pancreas or a kidney outside the body, to possibly replace a malfunctioning one, and to replace a malfunctioning one by transplantation. So this is the idea. This is the hope, that organs could be made from the cells taken from an arm, let's say, skin cells, reprogrammed into stem cells, and then grown into a different organ type. Now, to do that, you have to know how to guide the induced pluripotent stem cells, the iPSCs, towards the right fate. And this might involve giving them an extra dose of other genes, of the genes that are particularly active in the target tissue type that you're after. It turns out that making neurons from stem cells is rather easy. It seems that's almost like a sort of default state that these cells might acquire. So they just need a little nudge. But what's extraordinary is that if you do that, they don't grow into some random tangled mass of neurons. Left to their own devices in a dish, in a petri dish, these neurons will start to organize themselves into structures like those that appear in the brain itself, into, in fact, what are sometimes called mini brains. And here is a picture of one of these mini brains. And you can see there's an extraordinary amount of structure here. The different colors here are just different sort of stains that show different cell types that have appeared in the structure that is grown in a dish. Now, this is not my mini brain. It wasn't quite as beautiful as this, but this is my mini brain. This was what was grown from the skin cells taken from my arm. And again, the different colors show the different cell types, and the red ones here are neurons. And researchers are now finding ways of improving their ability to grow these mini brains, to supply more of the cues, the signals that in a normal embryo the surrounding tissue would give the cells, so that they can get them to be more brain-like. And these organoids, as they're called, these mini brains, can allow researchers to look at all kinds of questions, such as what can go wrong in normal brain development to produce abnormalities or pathologies like schizophrenia and epilepsy. For example, Madeleine Lancaster in Cambridge is using brain organoids to investigate how the size of the human brain gets fixed. And she studied microcephaly, which is a growth defect that results in an abnormally small brain size, and which can appear as a result of infection of a mother during pregnancy with the Zika virus. Now, my own mini brain was grown at University College London by Selena Ray and her colleagues Chris Lovejoy and Charlie Arbour. And it was grown as a part of a project called Brains in a Dish. And this was led by Charlie Murphy, and it was one of many, who is an artist, and it was one of many strands of a much larger project called Created Out of Mind. This was a project funded by the Wellcome Foundation between 2016 and 2018 to explore and challenge public understanding and perceptions of dementia. And Selena's team cultured the organoids from skin cells taken from my arm, and Charlie actually also had this underwent this same process. We both had mini brains grown, and the idea was that we would just see what our responses were to undergoing this process. Selena normally does this for people who have a genetic predisposition to early onset Alzheimer's disease. And so what she's hoping to do is to, by growing the mini brains, is to understand what goes wrong in the developmental process that might lead to that neurodegenerative condition appearing relatively early in the lives of those people who have that genetic predisposition. But brain organoids can be used in many other ways. They're probably the most evocative form of organoid that can be grown from iPSCs, but actually there are many other types of organoids that can be grown too. In principle, it's possible to use induced pluripotent stem cells reprogrammed with Yamanaka's cocktail of genes to grow any type of tissue and many other types of organoid. And this is actually, let me first of all, show you the process in which mine was grown. So these are the fibroblasts that were taken from my arm. These lumps here are actually pieces of my arm tissue, pieces of my flesh, and you can see the cells growing in culture here, streaming out from this this process. And this is what the brain organoid actually looked like in the petri dish at the end of that process. So here are some of the other organoids that can be grown from induced pluripotent stem cells: lungs, stomach, gut, all sorts. And you can see that many of them also start to take on the structures of the organism that would normally develop in an embryo. So, for example, in intestinal organoids, you start to see all the tiny protrusions called villi that appear in a normal embryo and that are responsible for absorbing nutrients from food. Now, already some of these organoids are being used as model systems to understand fundamental questions about how tissues and bodies grow. But as I said earlier, it's possible that they might also be used as replacements for parts of the body that aren't working properly. You could, for example, imagine replacing a damaged kidney with one grown from the patient's own induced stem cells. And what's more, making an organoid from stem cells means that you might be able to correct genetic defects along the way, so that if something is wrong with a person's organ because of some genetic mutation that they carry, you might be able to use gene editing methods to put that right, and then to grow the organism without that defect and then transplant it back. And if you're looking to regenerate damaged tissues this way, you don't necessarily need to grow whole organoids first in a dish. Yamanaka, for example, and others now are involved in experiments to inject retinal cells, so cells of the retina, of the eye's retina, grown from iPSCs, into the retina of patients suffering from macular degeneration. This is a condition that can cause blindness, and the hope there is that these cells will regenerate the damaged retina. And there are trials underway to inject iPSCs also that are on their way to becoming neurons, to inject those into the brains of people who are suffering from neurodegenerative diseases like Parkinson's, to see whether a healthy brain tissue could be regrown, or to inject them into the spinal columns of people who've had a paralyzing injury of the spinal column. So it all sounds very promising, but there are some big challenges. For one thing, transplanting tissues grown outside of the body back into it doesn't always work very well, because sometimes those tissues have to be very sort of precisely integrated into the existing tissues that are there already. So, for example, if you're trying to regenerate neurons in the brain or heart muscle to treat heart disease, then it's no good growing a bit of heart muscle this way and then sort of trying to stitch it back onto the heart, because it won't necessarily then beat in synchrony with the rest of the cells. It has to be integrated. And one option in a case like this is to, instead of growing them outside the body, is to actually regrow the tissue in situ, directly inside the body, by reprogramming the cells there. Now, there are hazards associated with this. One of them is that if you turn cells inside the body into stem cells, there's a danger that, because they're very good at proliferating, that they could instead develop into cancer cells. But reprogramming cells doesn't necessarily mean having to take them all the way back to a stem cell state and then growing them into a different tissue type. There's now a lot of work into what's called direct reprogramming of cells, where instead of taking them, here's Waddington's Valley again, instead of taking them back up the valley and then down another channel, what the aim is to just kick them straight directly from one valley into another, to transform them from one mature tissue type into another, a skin cell, say, into a heart muscle cell. And this too is done by injecting the genes that are highly active in the target cell into the initial cells, and hoping that that's going to be enough to kick it into a different cell state. And there are now trials underway to try to treat heart disease this way, and to see whether it might also work for spinal column injury. Another hitch in using organoids for regenerative medicine is that they stay little. My mini brain, I mean, they're called mini brains because they are very, very small. They're about the size of a dried lentil, a few millimeters across at best. And they can't grow any bigger than that, because if they do, the cells inside that mass of tissue lack a blood supply. They lack any access to nutrients and oxygen, and they start to die. Now, there's now some work aimed at getting around that problem so that you can grow them bigger by making blood vessels that will be able to grow alongside the development of the rest of the tissue. And some people are trying this using a kind of artificial scaffold to grow the cells in that already has channels sort of laced into it that can act as a blood supply. But perhaps the most promising approach to making organoids bigger with a blood supply this way is to grow them not in a dish, but inside another organism, where blood vessel networks will tend to develop naturally. And this is where things get controversial, let me warn you. For example, a Japanese group has grown liver-like organoids from human-induced pluripotent stem cells in a dish. So they're just normal little organoids. And then once they get to a stage of being what are called liver buds, so they're starting to look like little livers, then they're transplanted into mice. And there, the stem cells, the human stem cells, get the signals from surrounding tissues that say you need to grow some blood vessels. And so they start developing blood vessels. Blood vessels start colonizing that organoid. So they're human blood vessels supplied with mouse blood, growing a human organoid inside the mouse. And organisms that have a mixture of cells from different genomes are called chimeras. They're genetic mosaics. And these experiments show that actually you can make chimeras that are trans species, cells of entirely different species, so long as they're not too far removed, and it seems that humans and mice are close enough, they can get along just fine in a single organism. They can survive. Now, of course, you're not going to be able to grow a full-size human liver, let's say, in a mouse. But could you do it in a human-sized organism? Could you do it in a pig or a cow? Well, this too is being explored. In 2017, a team of researchers from California reported that human iPSCs can survive and continue to grow, although not terribly efficiently, after they've been added to the embryos of pigs and cows at the very early stage of the blastocyst. You put them in the blastocyst and they will continue to grow. But the question is, well, how can you get them to grow into, if you want a human kidney, let's say this way, how can you make sure that they grow into a kidney? And the way that's done is that the pig embryos, for example, can be gene edited so that they lack the genes that are needed to grow a liver. And researchers say that this creates a niche within that organism for the human organ that you want to grow. So it just then it's just the cleverness really of the embryo that as it grows, there comes a time when it needs to grow a liver, and the embryo, it's as though the embryo says, right, it's time to grow a liver, but we don't have the genes to do that. Oh, but here are some other cells that do have the capability to grow into a liver. These are the human cells, so we'll use them to grow the liver. This is what this is the idea of what might happen. Now, the medical value of this, of being able to grow entire human organisms this way for transplantation, is enormous. It could potentially save many lives. And if you're rearing livestock like pigs for human consumption, to make pork and bacon, to put it bluntly, why not at the same time grow human organs and harvest them for transplants, so long as the well-being of the pig isn't impacted? So that that's the ethical argument for. But all the same, I think I'm probably not alone in thinking that there are some very big ethical challenges here, and that our gut response kind of rebels against it. The word chimera, as I said earlier, it's a technical term used now for an organism with different genomes in some of its cells. But of course, it comes from mythology, and it refers to a monster. And a pig with human organs does sound pretty monstrous. And I hope it's not merely sensationalist to say this, that I have actually heard experts in this field admit that it's not obviously impossible in principle to grow a pig, not with a human liver or kidney, but with a human brain, fully sustained by a blood vascular system. Now, there's absolutely no reason why anyone would want to do that, no good reason why anyone would want to do that. And there are clear legal and ethical reasons why they cannot do so at present. But I want to raise the image, not to shock you, but to show just how profoundly these new discoveries in cell biology force us to reconsider what being a human being actually means. And now let me take you in another direction that explores that issue. Because of all the tissue types that can be made from embryonic and induced pluripotent stem cells, one of the most challenging and controversial are the so-called gametes, the reproductive cells, eggs and sperm. It's harder to do this than for most other tissue types, and that's because gametes are rather special. They only contain one copy rather than two copies of each of our chromosomes, so that, of course, when the gametes meet, then they get their full complement of chromosomes, one from each of the sexual partners, one from each parent, and they develop into a human. So to get cells that only have half the normal number of chromosomes, that's a special process that happens during development. It's called meiosis. And it's not easy to recapitulate that process in a Petri dish. But making fully functional eggs from iPSCs is possible and has already been done in mice. Mitanori Sato's team at Kyoto has transformed iPSCs in vitro, in a Petri dish, into the precursors of gametes. These are called primordial germ cells, and which haven't yet undergone the chromosome halving of meiosis. So they're not actually really like eggs or sperm yet. But then Sato's team could finish that job off by transplanting those primordial germ cells into the ovaries of live mice, where the cells completed their development into mature eggs, and they could use those eggs to grow by mouse IVF to grow perfectly healthy organisms. So this is the process that they underwent, and these are some of the mice that were grown that way. And in fact, Sato's team could carry out the entire reproductive cycle entirely in vivo. So they found that they could actually not, rather than transplanting the primordial germ cells into a live mouse into the ovaries of live mouse, they could actually take some ovary cells and cultivate them in the petri dish alongside the primordial germ cells, and that would be enough. The signals from the ovary cells would be enough to complete that developmental process, and that way they could make eggs entirely in vitro in a petri dish, fertilize them, and start growing another embryo, and then use the stem cells from that embryo to repeat that process, and in principle make generation after generation without ever making a live mouse. Now, whether all this can work for humans is another matter, but many groups are working on it, and there's at least some progress towards being able to make in a petri dish, towards making these primordial germ cells. And then there's a question of whether you can then take it to the stage where you can actually get eggs that can be fertilized. The same thing can happen with sperm. It's basically the same process that you can make the primordial germ cells, which have not yet decided whether to become eggs or sperm, transplant them into the testes of live mice, and then those would develop into sperm that could be used for IVF, for mouse IVF, that were clearly healthy sperm that could be used to fertilize eggs. Lots of testing will be necessary to establish the safety of these methods if they're ever going to be used for assisted conception in humans. But experts in this field don't see any fundamental problem with repeating for humans what has already been done for mice. The implications could go well beyond providing eggs and sperm for people who lack them to solve infertility problems in IVF. Because if you could grow eggs this way, then at the moment, egg collection for IVF is not a pleasant process at all. It involves hormone treatments and a painful process of having to then extract the eggs for in vitro fertilization. If you could do this from a scrap of skin, to grow eggs from this, not only would it be painless and much easier, but you can make many, many more of them. In a typical round of IVF, you might get a handful of viable eggs, whereas this way you could make hundreds or thousands. With a technology like that in place, you could imagine the stage being set for widespread use of embryo selection, where you grow a lot of embryos and then you select the ones that have a particular genetic profile. This is something that's already done to avoid genetic diseases. And in fact, in the UK, this is the only reason for which it can be done. There are several hundred diseases, genetic diseases, for which this sort of embryo screening can be conducted to find embryos that don't have that mutation and use those for IVF. But if you could grow thousands of embryos this way, where does that process stop? If you can then genetically screen every one of them and find out not just whether it has a genetic defect or not, but as we are increasingly able to do, to make some kind of deductions about the characteristics of a person who might grow from that embryo, then you can see that there's the stage is really set for a different landscape of assisted reproduction. You can even consider more exotic permutations in how we've reproduced, because it's possible in principle for both the cells taken from both men and women to be turned into eggs and sperm. You can get in this way, in principle, you could grow eggs and sperm from either a man or a woman. And so there are all kinds of possibilities that you could imagine in theory coming from that. You could also take cells from people who normally couldn't reproduce, from very elderly people or from children or even fetuses. So you see that if this technology is possible, then there are some very, very challenging questions that are going to be asked. And now if you add to that picture the possibility of using genome editing in those scenarios, then you have a whole other set of questions. If you can then tinker with the genes, not just select which genes are already there, but tinker with the genes of embryos, then you're in another ballpark entirely. And for a long time, the idea that genome editing might be used in assisted reproduction was considered sort of it was considered out of the park. People were generally agreed that we are not ready to do this. But then, as some of you may know, at the end of 2018, November 2018, a Chinese scientist named He Jiankui reported that he had done this, that he had actually produced twin girls by using a genome editing technique called CRISPR. This is the most accurate gene editing technique that at the moment we have, to produce two baby girls that he said he had edited the genomes of so that they would have immunity, increased immunity to infection with the AIDS virus, HIV. Now, this was widely and rightly, I think, condemned as shockingly irresponsible, because we simply do not know enough about the effects of this genome editing technique at the moment to know whether it's safe to use, whether there might be long-term consequences, whether there might be consequences that appear later in the lives of people who have been born this way. So that work was widely condemned within China and within the international community, but it has opened up, in a way, it's opened up Pandora's box, because it has shown us that it's possible in principle to use this technique to create live births, and as far as we know at the moment, apparently healthy births. And so we now have to urgently debate under what circumstances that may or may not be permissible. All of this already, the idea of growing eggs and sperm from reprogrammed cells in the lab, already that suggests that we might be entering a new phase of assisted reproduction. But things could go even further than that. This is an example of what I mean. This is an embryo-like structure made from mouse cells. However, it was made without any eggs and sperm at all. It's an organoid growing in culture, except that this time the organoid is the entire embryo. You could consider it an embryoid. And this was made by researchers in Cambridge by assembling the different kinds of stem cells taken from embryos. And what I mean by that is that you might remember at the beginning I said that in an early stage embryo, there are several different types of cell. There are the embryonic stem cells marked in red here, which are the ones that are going to grow into the organism. And then there are what are called trophoblast cells, which are stem cells, which are the ones that are going to grow into a placenta. And surrounding all of this, the extra embryonic endoderm cells, which are the ones that make the yolk sac. And the researchers in Cambridge found that if you simply put these different cell types together, they will spontaneously organize themselves into this sort of peanut-shaped structure, which looks rather like an embryo just beginning to enter the gastrulation phase that I talked about, with this cavity in the middle. And some of the embryonic stem cells here already start to show signs of beginning to differentiate, as they do in a normal embryo, into the mesoderm. Now, all of this, so this really is an artificially made embryo. And these are mouse cells, as I say, but there is work also along these same lines using human cells. And one expert in this area, Martin Perra, has said there is no reason to believe that there are any insurmountable barriers to the creation of cell culture entities that resemble the human post-implantation embryo in vitro. In other words, that we might be able to assemble something that looks very much like human embryo this way. Now, the question is, how do we think about these entities? What sort of entities are they? They're clearly not embryos in the normal sense. And at the moment, there's no reason to think that they could develop into a whole organism. They may not have quite what it takes. But as research progresses, there's no reason to think either that they might not get to that stage, or at least to progress to more advanced stages of development, perhaps, entirely outside the body in an environment that mimics the uterus, that gives them something to implant into so that they can develop further. the biologist George Church of Harvard University has proposed that things like this, entities like this, be called synthetic human entities with embryo-like features. There's an acronym for that. It's SHIEF. And these are like nothing produced by conventional IVF, and it's unclear how to regulate them. I said earlier that regulation on human embryo research is limited by the 14-day limit. So you can't experiment on them for longer than 14 days because that's when this primitive streak begins to appear. But it's not clear that sheaths would even follow the same developmental pathway. They may not grow a primitive streak at all. We simply don't know what kind of regulation is therefore appropriate for these sorts of entities, partly because we don't quite know how to think about their ethical status at all. And this is just one indication that very difficult choices about these technologies are upon us, whether we like it or not. How we should handle those choices isn't obvious, but I think it's clear that it needs strong legislation, as we have in the UK under the remit of the Human Fertilization and Embryology Authority. We also need international guidelines and rules and an inclusive dialogue between scientists, ethicists, and many other members of society. This isn't just a matter of do's and don'ts, though. What we're dealing with here are questions that take us right outside our normal frameworks for medicine and research, and which pose, I think, profound challenges for our usual preconceptions and categories. We really are replotting the human. So here's my mini brain again, and this was discussed in a recent paper by the bioethicists Hank Greeley and Barbara Knoppers, who say that it's actually unclear what these technologies mean for the legal definition of what is and what isn't human. And these are questions that go, I think, even deeper than that, deeper than what sort of regulation we're going to use and what sort of things we're going to permit and forbid, because I think they challenge our whole view of who we are. We have a sense of being autonomous, self-contained beings, of course, but I can tell you that having had a mini brain of my own grown from a piece of me across 5 miles away across town has put a little seed of doubt in my mind about that notion of identity and self. So does the notion that my stem cells, which still exist in cold storage somewhere, some of them are still there, they might in principle be grown into eggs and sperm and embryoids. What does that mean for my own sense of myself? Once we really come to terms with the fact that we are all communities of cells, including many cells like our gut bacteria that don't even have our genomes, then I think it becomes no longer clear where our boundaries, our narratives, indeed our lives start and end. We believe that we're individuals, and yet biology can offer no rigorous definition of what that means. So it's an unnerving and exhilarating message that our cells are sending us, and I think that we need to start listening to it. Thank you.
Thank you very much, Phil. That was really fascinating and really nicely captured the issues that are covered in your book. So I'm going to kick off with a few questions to give us a little bit of a discussion, if that's okay.
Sure. Yeah.
I actually want to there's several things I want to ask you about this. It's so fascinating. But I want to start with a big question, which you hinted at a little bit at the end. A lot of your book talks about the very early stages of human life, or perhaps what happens before there. And you referenced a couple of times in your presentation the 14-day rule for experimenting on embryonic cells. And there's a clear sense that from the research that's happening now, that there's something slightly arbitrary about that 14-day rule. I mean, there's a moment, the appearance of the primitive streak. But I'm also interested to know how this idea of ourselves as communities of cells affects the other end of life, affects death. You mentioned in your presentation that cells need access to nutrients to stay alive. And clearly, there is a process that happens at the end of a human life which leads to the cells in our bodies dying and the bodies decaying. But it's a long process. Humans have always tried to capture that moment of death and being able to decide whether someone's whether someone's alive or dead. We've looked at breathing stopping. There's traditional holding a mirror in front of someone's mouth. We've looked at the heart stopping, but both of those are reversible. Don't seem to capture our idea of death. More recently, we've picked up on the idea of brain death, but it's not even clear that any of the things we measure when we talk about brain death are irreversible. So what I'm interested to know is, does what you're telling us about humans being fundamentally communities of cells also have some implication for the idea that death is very different, and that we may never be able to find a criterion of irreversible dying?
Yeah, well, that's a great question, Tom. And, I think that in the end, that too is probably a philosophical rather than a biological question. And I think in some ways it's related to the question of the beginning, of when life begins. Because we talk about this notion of when life begins. What I say in the book is that actually what we're really saying is, what we're really trying to pin down is, when is an identifiable individual, when does the self begin? Because life never ends. Life has never ended since it began maybe 4 billion years ago on Earth. It's simply been passed on from cell to cell. So there is no question of, there is no real question of when life begins. It's about when the self begins, and the same will be true of when the self ends. And clearly, as you say, the death is a complex process. Death isn't a moment, and we're always pushing back that sort of boundary of, when in the past people might have been pronounced dead, perhaps, today there are ways of recovering them. We don't know how far that can be taken. But if we think about, for example, this notion of regenerating organs, then you get into the philosophical question of Theseus's boat. If I remember, it was Theseus, wasn't it? That yeah. where the boat was continually repaired, and the question became, well, is it still the same boat if you've had every plank in it replaced? And we're starting to have to think about that possibility for humans. If you can regrow all of these organs, and if they perform, as healthy organs rather than as aged organs, if they've been turned back to stem cells and then regrown, then you'll be having, if you like, a youthful liver replacement, perhaps. And how far can you take that? How far can you keep the human body going that way? And I think we probably have a sense that, well, as long as our brain is the same, then it's, that's going to then we're the same person, even if the whole body has been replenished. But even the brain, as I say, is kind of, if you like, up for grabs, because neurodegeneration is the big issue in aging now, and now that we're living longer and longer. If we have methods that will enable the brain tissue that might be disrupted by neurodegeneration to be regenerated, it won't have your same memories, but it will. The hope is that it will give you cognitive facilities that you would otherwise be losing, the ability to form new memories. So is that the same self? If your old memories are gradually fading, but you're getting new ones, where's the person in that? And I think these are questions that they may be irresolvable. And in a sense, I think they're irresolvable because of the perhaps incompatibility, certainly the disjunction between our lived experience of having this integrated whole and this being somehow the self, and the biological reality of our flesh and blood being a collection of cells. And we don't quite know how to bridge that gap. And it's actually one of the astonishing feats of evolution that this community of cells somehow evolved to create this sense of unity. We don't know how that happens because different senses, register at different rates. We're having sensory input all the time, and somehow the brain integrates that into this unified sense. But if the brain itself is being altered and regrown, then we don't quite know where that's going to leave us. So I think it is a very striking, and it's going to be a very real question that we're going to have to contemplate of, where in all of this biological activity, this cell activity, this formation of organs, this activity of neurons, where is the self?
Fascinating, yes, and touches on lots of philosophical questions. I could now advertise, come and do a philosophy degree, but rather than do that, I just want to pick up something you mentioned there, which relates back to something very interesting you said in your presentation. You talked about neurodegeneration being one of the new medical problems that we're getting as people live longer. But you also mentioned in your presentation that when cells are being grown in vitro, that it's actually easiest to generate new neurons. So I wondered, one, first of all, why? Is there some interesting reason why it's easier to trigger the development of new neurons, why we get mini brains? But also whether that has any tension with the thought that we discover neurodegeneration is an issue.
Well, the answer to the first question is not really one I can give you, I'm afraid, although it's one I asked myself. I was very interested when my cells were being grown into neurons. I was, saying, well, what exactly are you doing? I'd imagined that it was going to involve the addition of genes specific to neurons that would trigger this, but but it didn't. That wasn't necessary. It was just a little tweaking of the nutrient medium. So it, just turned out to be very easy to do that. I mean, I should say another common state that cells left to their own devices, stem cells would differentiate into fibroblasts, which is the sort of connective tissue in skin. That's another default state. So I don't know if anyone really understands why it is that this seems to be one of the default states and not, say, kidney cells or whatever. An interesting question for me about that is whether there are evolutionary reasons why that might be so. It seems to suggest that perhaps neurons were sort of fundamental to very early organisms, and whether there's a sort of recapitulation almost of the evolutionary process that's going on there. I don't know whether that's so or not. In fact, the whole question of how nervous systems evolved is itself a fascinating one and a very complex one. But it seems clear that they once you started getting complex many-celled organisms, you needed a nervous system for various things, for locomotion, to control different parts of the body, but also for perception. And it may be that those two things were separate. So there are all sorts of interesting issues about where nerves came from in the first place, where nervous systems came from. In terms of neurodegeneration, I guess your question was that if it's so easy, then why doesn't it happen in the brain? It does actually happen in the brain. Actually, the new neurons. It used to be thought that new neurons don't grow in the brain at all, but quite recently it's been shown that actually they can, and they do. Most of our tissues, in fact, have what are called adult stem cells within them, which aren't pluripotent like embryonic stem cells. They can't do everything, but they can grow into various, related tissue types within that particular organ. And that happens in the brain too. So there is some capacity to regenerate neurons there, but it's very, very limited. And it's certainly not enough to cope with the kind of massive neuron death that happens in some of these degenerative conditions like Alzheimer's. So that's really the problem that we're facing there. And there was something I was going to add about the question of no, I'll leave it at that. That's
really interesting. I'm slightly relieved that we do manage to regrow some brain cells. Gives me a little bit of hope.
It doesn't feel like it, but yes.
That's great. So one last question. I must admit, this is the first time I've done one of these pre-recorded events, and I'm loving the fact I can abuse my chair's position and ask all the questions I want to first. So I'm just going to ask one last one. So the tension you set up between our sense of ourselves as autonomous individuals and the biological conception of ourselves as communities of cells. I like that phrase, communities of cells. You see this as problematic because there isn't anything within biology that maps particularly well onto that sense of ourselves as individuals. And I kind of wondered why you think biology is the place we should be looking then. Perhaps what we're learning is that what we talk about when we talk about ourselves as humans isn't actually biological entities. It's some other form of, I'm not suggesting non-material, but I'm thinking a different way of thinking about and conceptualizing what's there in the world. We think of ourselves perhaps not as animals, but as a different type of being, which is also an animal, but the self is something slightly different way of thinking.
Yeah, well, actually, I agree with you that I think biology is probably not the place that we should be looking to develop some kind of conception of what we mean by the self. And it worries me that not just biology in general, but specifically genomics is becoming increasingly the sort of locus of where our self is supposed to be identified. And you see these companies like 23andMe that offer to sequence your genome, advertising as, find out about the real you. This is the real you. It's in your genes. It's in your genome. It seems to me is fundamentally misconceived. There is an awful lot in your genome that is that will make sense of the way we develop and the way we look and even the way we act. It's undeniable that your genome has an effect on the way you behave, but it's not all of it by any means whatsoever. I mean, it's clear that it can't be. There are the number of connections of neurons in the brain, the number of synapses, astronomically exceeds the amount of information that's in a genome. It can't prescribe it all. And we know very well that as the brain develops, there's a lot of chance involved in how things wire up. There's a lot of noise involved, and out of that will come certain kinds of predispositions and so on. It's not all genetically encoded, and beyond that, even it seems to me that the question we've often tried to pose of when does life begin, when does the human begin, and how in that case should we decide about things like abortion and IVF and embryo research. It troubles me that has come down to biological questions of gastrulation and fertilization and so on. It's necessary that it did, and I think it's good that in this country, at least, we were able to use that to develop some pragmatic regulations for embryo research. But that's not about when personhood begins, because it seems to me surely personhood is something that emerges by degrees through the lived experience. And we know that our brains are affected by that. Our brains are remolded by the experiences we have. Then, it's not as though there's just that any of that is set in stone at any point in our development. And all of that will affect our experience and our sense of self. So I agree. It seems to me that when we start to think about issues of self and personhood, we have to take into account the lived experience, as an entity that is guided by, to some extent, our inherent biological disposition, some of which is in our genes, but is by no means determined by that or defined by that.
Thanks, fascinating answer. I'd love now to go on into a discussion about whether we're making mistakes tying this notion of self also, or using it to ground our ethical decisions, and that actually you've just hinted that personhood might be something that develops, maybe something that develops in early childhood, but that doesn't mean that we only have moral responsibilities to what those beings we regard as fully persons. But that's fascinating. I mean, now I've raised that, I better let you say something about it.
Well, I suppose what all I would say about that is that these are questions, one can sort of sit and talk about them philosophically, and it's lovely to do so, but actually they're going to be very practical. Increasingly, they're going to be practical questions. The onset of IVF, the invention of IVF, made that very clear. It made us really have to think about these questions of, whether we can define a moment when personhood begins, and how we can frame regulations about this research. But once we start getting into the possibilities, of genome editing, for example, and of new methods of reproduction, and we've had these discussions with, for example, the technique of mitochondrial transfer, where in order to avoid certain diseases of compartments in cells that can be very horrible and that some people have a genetic predisposition to, we take those compartments from a third person, in effect, and add them to, it doesn't quite work that way, but add them to a fertilized egg, so that you have some genetic material, because there are a few genes in that compartment, you have some genetic material from a third person. And we've talked about, and this gets talked about as three-parent babies, and there's a sort of furore about how unnatural that seems, but actually it seems to me that the controversy about that, quite aside from safety issues, which are always important to look at very carefully, the worries we have about that are really about this incongruity between our kind of lived notion of what it means to be a parent, and what it means to have a certain sort of heritage, and what our biology tells us about that. And some of the more outlandish possibilities that I touched on in my talk, and there are ones that go further still, I can assure you. They need. I think it's good to frame questions like that so that they can really confront us with the challenges that we face. Not because anyone thinks that it's a good research agenda to follow, but because they really force us to show that, in some ways, they force us to recognize that we can't root our answers and our understanding of those sorts of issues, of our experience, our personhood, ourself. We can't just look to biology to answer them. We're going to need to find some other sort of framework to talk about them, and it's a framework that goes beyond any we have already, because the kind of permutations, the kinds of possibilities for starting life, for affecting and transforming life, and, as you say, perhaps for ending life, those are all changing in ways that have never happened before. So we're going to have entirely new ethical and moral questions and philosophical questions that we'll need to grapple with.
So, Phil, thank you very much. Fascinating presentation. Thank you for letting me abuse my position as chair and ask you those questions, but they're really, really interesting stuff. So it's been a great talk and a great evening. I just want to let the audience know that Phil will be on Twitter straight away after this, answering, or trying to answer in those short gaps, some of your questions. I also want to let everyone know that for the Festival of Ideas, we've again teamed up with local bookseller Fox Lane Books to provide autographed copies of the books that are being discussed here. So Fox Lane Books is currently working online, and you can find their website at www.foxlanebooks.co.uk, where they have a special Festival of Ideas page, and you can buy a copy of How to Grow a Human with a signed book plate. And they're also selling one of Phil's other recent books, Beyond Weird, on quantum mechanics. So before I do the traditional way of demonstrating our thanks and our gratitude to Phil, I just want to say that we hope everyone who's been listening in will continue to be engaged with York Festival Ideas online. You can check out the website, yorkfestivalofideas.com, for full details of all the other events in the programme this year. And we'd love to hear your thoughts on these events and to carry on the conversation. And if you're using social media, please use the hashtag #YorkIdeas. So Phil, thank you very much indeed. Fantastic evening.
Thanks very much.